For our expert panel, I have three new faces to introduce. So Dr. Chiara Heide is the founder of Bright Cure based in London. Dr. Oliver Worsley is co-founder and CEO of Sequential Skin based in the UK and the US. And Dr. Ryan Keane is lecturer in clinical microbiology based at Glasgow Caledonian University. And Ryan won NBIC proof of concept funding in this area as well. So he will speak about his project. I've asked you to have very brief kind of introduction of highlights, so only 3 to 5 minutes, please. And we can start with you, Chiara. So if you want to share the screen, please start.
I hope everyone can see this. I can put it to full screen mode as well. Perfect. Yeah, so hi everyone. I'm really happy to be here today. I'm Chiara. I'm the founder and CEO of Bright Cure. Bright Cure is a biotech company, and we're developing next generation microbiome products for women who are suffering from recurrent urinary tract and intimate infections. I have suffered, so I'm a scientist. I did my PhD at Imperial, actually in chemical engineering, bioengineering, synthetic biology. But I founded Bright Cure because I personally suffered a lot from urinary tract infections and was just like really frustrated with solutions in the space and constantly taking antibiotics. However, I'm actually not alone, and I think many of you know that UTIs are a very common infection, and women, actually every second woman suffers from it by the age of 32. And then a lot of women like me end up constantly, taking antibiotics, being stuck in this painful cycle. And obviously then the antibiotics also have a lot of other side effects with like fungal infections and destroying the vaginal microbiome, not only the gut microbiome. So frustrated with the current treatment situation and also the lack of awareness in the space, I founded Bright Cure after I finished my PhD, and we're really bringing a completely new mechanism of action, new technology to the market. Unfortunately, I cannot say too much about it here, but it's not another probiotic and it's not another cranberry supplement. It's really a new technology we're bringing to the market, and technology will be, it's a live organism that we will package into an intimate cream, and then are planning a whole product range around a whole product range for women to kind of address the specific issues in the life cycle of women. To give you a bit of an idea, because Katerina asked me to kind of give you an idea where we are. So we basically started or got our first pre-seed investment from NBIC, which is this big biotech accelerator in the US. The money from NBIC really allowed us to do a proof of concept, to get our own lab, which we have now in London, and we filed a patent then as well. And then we just recently raised another seed round, which was really exciting with First Minute. There's some UK investors. Mitchell is a fund from Peter Thiel, Energy Rorian, and Calm Storm. And that's really now to move the technology out of the lab. So we're now moving it into manufacturing. So we're moving the ingredient into manufacturing, and then also doing our first trials, which we are really excited about. We hope that we can then launch the product kind of summer next year, probably autumn next year. And I think also I was supposed to talk a bit about the challenges I faced and quickly about the contribution that we add to the women's health space. I think for us, one of the main challenges was also that we started from scratch. So I finished my PhD, I had this idea, I really wanted to address this problem. So I really started with this problem and looking into different technologies to address that. But that meant that we didn't really have any data at the beginning, and we were not really like a university spin-off, so this has nothing to do with my PhD. So I think it was quite difficult to initially find the right lab space and get the funding without having any proof of concept. So really starting from scratch, I think, was really tricky, especially because it's not a tech startup, so you cannot just sit with a laptop kind of in a Starbucks and produce a proof of concept. And I think also funding was tricky because we're kind of at the interface of innovation, of kind of like biotech and consumer. So we're launching this as a consumer product, but it's quite a lot of innovation. Innovation is costly. People don't really like to fund R&D, so I think that was a bit tricky. And then I'm also a solo female founder, so I think a lot of people then don't take you as seriously. And I had some people who are not saying too many nice things to me. In terms of contributions, and I think this is the more positive part here, I think is that we're bringing innovation into the space, so really offering a new solution for women, like a healthy alternative, hopefully, to antibiotics. One really key aspect for me was also that I wanted to empower women to take control of their own intimate health and not have this barrier to kind of access solutions that are scientifically proven and that work. And another one is really like a more broader one, because this is obviously also a platform technology that we are keen to also look for other applications into the market, is reduce generally the overuse of antibiotics and obviously all side effects and problems that it brings to our society. So I'm really happy to be here today. Thanks for having me. And yeah, you can also reach me via my email.
Thanks very much. We will answer more in questions. Oliver, I've asked you a little bit to look at the mother-infant biome transfer, but if you want to say a few words about your company first, please.
Yeah, sure. I have a few slides as well if you want me to.
Okay, share them then.
On through those. Yeah, let me just. Okay, just checking you can see the screen.
Not yet, but in a minute we will. Yes.
Okay, so hi everyone, my name is Oliver. I'm the co-founder and CEO of Sequential Skin. We're a testing skin microbiome company. We started out originally by this idea that we could collect both your genetics and your skin microbiome all in one shot. And we know that conditions like atopic dermatitis involve so heavily both components, so we wanted to make sure we captured these things. We ended up creating a kit. It's a really simple kit. It contains an adhesive patch. It can be done from a volunteer's house, a patient's house, consumer's house. It comes back to us in the lab when we do the downstream analysis. So very simple to use. It takes about 10 seconds to take the sample. You are wearing a pair of gloves, but this is just for kind of effect. And we find that this is an improvement over a swab. So we don't have the same variability that we do with the swab. This is more comfortable for the consumer or patient. And it's also, we get things like increased biomass with DNA. We're also seeing we can get increased integrity of RNA when we recover both samples from the swab or from the patch. So yeah, we're seeing really interesting things, especially on the diversity side, where we can correlate certain traits of skin with diversity. This is a study we did in Asia. We're seeing some correlation between redness and sensitivity and the diversity of the skin microbiome. We're also working with a lot of skincare companies to understand more about the products they use and how that affects the skin microbiome. And so this is an example of kind of a 30-day study, as well as longitudinal studies that we're doing now, where we can see that certain products are interestingly affecting the skin microbiome. So working in more of an in vivo setting. I think relevant to this conversation, and excited to talk to all of you, is more of the clinical studies we're doing. So we have a really interesting study on atopic dermatitis in infants. We're going down to the age of 1 month in these patients. And we've developed a sequential skin kit, which I can show you. I've got the skin patches here as well. They're smaller patches, and they've been optimized for use in infants. And we're getting some kind of early data that I can share, and yeah, looking forward to having that discussion. But the team itself is myself, Albert, and Petroni. Petroni is our skincare director and pharmacist. Albert is our chief scientific officer and has worked in genomics. And we've got an amazing set of advisors, both on the scientific side that are expert in skin microbiome and on the dermatology side. So yeah, thanks for having me, Katrina.
You're very welcome, and you will have more opportunity to obviously elaborate in the questions. And Ryan, can I ask you to share, please?
Can you see that okay?
Yes, we can, thank you.
Yep, excellent. Yeah, so first and foremost, thanks very much for inviting me to present our project, which was kindly funded in the POC4 call. So here we're looking at evaluating the in vitro efficacy of a novel antimicrobial against polymicrobial biofilms, which we will form in vitro in the lab. And this is basically just our overall project workflow. So we have two outputs, or two main outputs from our project. So first and foremost is demonstrating the efficacy of our novel antimicrobial, which has been designed by our industrial collaborator. So the key thing about this antimicrobial is it's species specific. So it's a very, very targeted approach to targeting these pathogens which are found within our BV model. So our BV model itself is informed based upon microbiome data, and it will contain about 8 different bacterial species, and these will be split into both pathogens, so pathogens of interest, such as Gardnerella vaginalis, which we're hoping to target with the antimicrobial, but the biofilm model also contains some of these commensals, and particularly these desirable lactic acid producing lactobacilli. What we aim to do, and this leads us into our second outcome from the project, and that we want to be able to design an analytical tool where we're able to basically evaluate and quantify any shifts and changes within our biofilm model. So if we're able to target a specific pathogen, we wipe out, we kill this pathogen, what then happens to the kind of good or the healthy component of the microbiome and these desirable lactobacillus species. So to do that, we're going to use a technique which is called quantitative microbiome profiling. So this is something which has been used and published before, but predominantly in clinical studies of clinical microbiome data sets, whereas we're looking at it in terms of a mock community or an in vitro grown community. And this technique can basically overcome some of the hurdles and caveats of traditional microbiome sequencing. And with this traditional approach, you get a relative abundance. You're not able to distinguish between live and dead cells, so you don't actually get an overall quantity. Everything's basically given as an abundance or a percentage of the full community. But what we can do is within our analysis pipeline and experimental pipeline, we factor in 2 additional experiments. And this is using FACS-based quantification, which is a type of flow cytometry, which is able to differentiate between different cell types. So we're able to differentiate between if they're live or dead, and using a second method, which is Propidium monoazide-based quantification. So PMA is a molecule which will bind and collate to DNA. However, it's not able to cross the membrane of cells. So we take these 2 cells here as our bacteria. So our live cell here in the green, the PMA can't cross the membrane, it doesn't bind the DNA. But with our dead cell, which is its membrane compromised, it's able to get into the cell and bind to the DNA. And by binding to the DNA, it then inhibits any downstream processes. So these are things such as PCR and sequencing. What we then do is just the same as a traditional microbiome workflow: extract the DNA, prep our libraries, we can sequence it in my own lab using the nanopore. We then go on to do our QC and filtering our reads. But then what we can do is we can filter in this data here from the second part of our work stream and integrate it with the microbiome and sequencing data which we generate. And what our end output will be is that we're actually able to get a quantity of exactly how many cells are within our biofilm community. So if we have our treatment, does this then diminish our Gardnerella, and does it then promote the growth of some of our desirable bacteria? So that's a whistle-stop tour of our project. And again, just like to thank and acknowledge our collaborators, so both academic and industrial. Thank you.
Thank you very much, Ryan. So I will ask all the other speakers to come to the panel, please. And I will hand over to Jill to lead the panel. Thank you.
Thanks, Katerina, and thank you to all the speakers. Judging from the comments in the chat, people have found it very informative. One of the reasons for doing this is that we felt that the industry was using microbiome claims, but the science base in the UK perhaps wasn't strong enough yet to be able to underpin future commercial activities. And I think what we've learned today is that the science base is alive and kicking. I've learned a lot about what's going on before, you know, we organised this webinar. So it's really important, I think, to have a community of researchers working in this area. And Julie and I have just submitted a BBSRC networking grant, which is to build a community of skin microbiome researchers in the UK. So if they fund us, we'll be in touch. And if they don't fund us, we'll still be in touch, because we really think it's important to network everybody together. Anyway, I'm going to pick some questions from the chat and the Q&A. If you have any, for the people who are attending but not presenting, if you have any questions, please do put them in the Q&A. So some really interesting questions. One of the things that I think is really important is to understand, because I started the day talking about sampling. We do have bacteria and maybe fungi that live in the skin. As on the panel, like to comment on the implications of sampling bacteria and perhaps fungi and fungal spores that are not immediately available from a skin surface swab. Just open your microphone and reply if you have an answer to that question.
Yes, Jill. Just to add, I don't know if you're breaking up a little bit, but we're doing something where we've collected biopsies and our skin patch, and we're collecting the two of individuals to see the difference. We've got some early data to show that there is a good representation using the patch, which is representative of the biopsy as well. But it's kind of early to say, at least on our side.
Okay, thanks. From Jane Everson, part of the CCUK. This is amazing science, she says. How can the cosmetics and personal care industry take advantage of it? I think they already are, is the answer to the question. But are we into issues to do with modifying the microbiome?
Shall I reread this? You are cutting out a little bit. So again, Jane Everson is asking, how is this borderline pharmaceutical science, or can pharmaceutical industry space, or can the panel foresee cosmetic applications developing? Who wants to start, please? Kelly, I'll pick on you.
Sure. Yes, the answer, as far as I can tell, is that there's already people taking advantage of this. We work with some companies who are making probiotic skin mists and applications that you can use just by applying a cream that have, these beneficial microbes that are found like around the world, not just necessarily in the places that we're familiar with. And with the strain level analysis and with being able to grow these up in the lab, you can definitely imagine how this is going to be going a lot. We're going to see a lot more of it from the big pharmaceutical companies going forward.
David, what about you? You must see some of the companies.
We test postbiotics. All our clients are producing this sort of products. So I suppose our issue with the regulation and regulatory. So there are lots of probiotics for the guts. We eat yogurt every day. It's not a medicine. So it's an issue of regulation, how we are going to get those standards. First, regulatory issue of what a probiotic is. Can you put probiotics on your skin? People is doing this. And then is how you prove that those probiotics do what you're saying they're doing. So we need to reach a standard that tests those products from the cosmetics. The same way we test for irritation, the same way we test for sensitization. And it has to be an in vitro test, and it will probably then follow up with an in vivo test. And what we do in LabSkin is basically trying to create those standards for in vitro testing. And the industry is already doing that, trying to avoid to fall into a pharmacological effect. And that's what cosmetics always do. And we keep doing that until the regulatory issue comes in, and that's what we're working on to try to work on that regulation.
This is Marnie. I'll just add to that, is that the approaches such as you described, David, and we have similar ex vivo human skin models. I mean, I think also in the cosmetic industry, they want to develop technologies that actually have the desired effects. So that's how they're screening their chemistries and creating their IPs. And then you're right, once you have that data, where can you take that? Right now, the regulatory is not in place and you can't make a claim, so then it becomes supportive material, but it is being done.
So thanks, and I think including regulatory is probably very important in developing the science, so that the acceptance is something which will be carried along with the science for when new products come to market. Another question that came up is why is there such a high incidence of bacterial vaginosis in women? Does anybody have any suggestions?
I don't know if there's good, I mean, I don't know, Kelly, if you want to weigh in on that. It's multifactorial.
Yeah, I think there's so many conditions that can disrupt the vaginal microbiome. There's just general day-to-day activities. There's antibiotics, there's sexual, whether it's different partners, whether it's, you know, a sexually transmitted infection. There's just over the course of a woman's life, there's a very, very high chance that she will have an experience somewhere where she's going to have this problem. And then I think for lots of women, once they have it, it disrupts things for a long time. And also you get the hormone changes and just so many things like that.
Yeah, maybe to add here as well, because I have obviously like a lot of contact with like UTI patients as well. It's like a lot of women who suffer from UTIs, then obviously you start taking antibiotics, and then as you just said, Kelly, as well, then you know there's so many different life situations. When you become obviously sexually active, you have different partners. There's a strong correlation actually between, you know, UTIs and the bugs you're suffering from, also in connection with the penile microbiome, which is often completely actually ignored. It's quite interesting because often the women is targeted, or the women is seen as the problem, but it could be actually also her partner, which is kind of like reintroducing the bugs. And I think then when you start taking antibiotics, then you obviously disrupt your vaginal microbiome, and then you kind of keep on taking the antibiotics, and then you often also take antibiotica, a lot of women, because then they get fungal infections, and it's really hard, I think, to then get out of the cycle, yeah, unless you also then change your kind of sexual maybe behavior as well, or yeah, it's a big problem.
So Chiara, what's the role of education for young people in all of this?
I think a lot of women are actually already quite educated around this topic, maybe not so much on the scientific level, but kind of know what often triggers, you know, what triggers their infections, and then they're trying to avoid it. And I think a lot of women then also are trying to obviously adjust their lifestyles to it, which is quite extreme in some of the cases and quite sad in many ways, from the type of underwear they wear, or like sexual activities they have, or it really affects people's personal lives. So I think what is often though missing is also the whole connection around it. So it's not just UTIs, right? I mean, for example, the vaginal microbiome, if you then take a lot of antibiotics, it's related to obviously getting other infections in the intimate area as well. So I think not just looking so isolated at like one specific conditions, but I think this interaction of what it all triggers and how it's all connected, I think is really important. And I think it's often sometimes quite missed in the conversations. Like a lot of people focus on one thing, but actually it's kind of like all connected, because if you suffer from UTIs, you're more likely, you're more prone to potentially, sexually transmitted infections, because your vaginal microbiome is not so strong. You're more prone to get fungal infections. Then there's also very strong correlation with preterm birth, fertility, related problems as well. And if you're pregnant, you don't want to take antibiotics, but a lot of women suffer from UTIs a lot. So it's kind of like it's all connected. And I think it's really important to make people aware of this, connection, how it's kind of like all linked, and maybe give different options of how to break out of this cycle, basically.
Thanks very much. So Matthew Cummings asked a question again to you, Chiara. Are you able to produce live organisms at scale easily? Some bacteria are tough to produce in fermenters.
I obviously cannot comment, unfortunately, so much about this. But yeah, we work with very experienced partners who are helping us to scale the technology, because this is, we're obviously producing a new ingredient, a new active ingredient. I think with some it's definitely challenging, especially because they require specific conditions. And I think often also a stability issue is widely discussed, like in terms of delivering topical applications, including those live organisms. I think there's always a particular challenge around that a lot of people are aware about. We know that our active ingredient is scalable, but how well, I mean, we're still quite early stage, so how well we can deliver that, we were saying, fingers crossed.
Maybe just a question to those people who are using models. What's the problem with engraftment of introduced species to the models? So maybe those people who are developing the models, Ryan, David, could comment on that.
I'll maybe come in just now if that's okay. One of the things, so I did my PhD in a dental school, so we worked in the oral cavity and used a lot of representative models, and there certainly is a lot of R&D goes alongside it. So it's not necessarily, when we grow things in the lab, it's not adding bacteria A, adding bacteria B, and then seeing what exactly they do. There's a lot of kind of dynamics you've got to consider, things like early colonizers. So what we used to do in the oral cavity was that there'd be various different streptococci species, which we would have to put in first to establish the biofilm. And then every few days, we would then add in different pathogens. And what you then get is the end stage dysbiosis, where you'd have what would be representative of the infection you were targeting. And this is something which we are considering just now with our own project and our own model. It's not necessarily just adding everything into the mix and almost hoping for the best. There is not some skill, but there is some things you need to consider, particularly with early colonizers, and so you want to stabilize the community, particularly with BV, which we're working on because it's the anaerobes.
Thanks. Oliver, I noticed you were using the adhesive patch to collect your samples, and does the choice of the adhesive make much difference to extracting the sample? Because that's traditionally been quite an issue with using tape strips to collect samples.
Yeah, it is. We've spent about 2 years now to finding the right adhesives, so going through really hundreds of different adhesives to make sure we're collecting sufficient material, but done in a reproducible way, so consumers or patients can use it in an easy way to capture that information. So yeah, we have done quite a bit of work, and if anyone's interested in trying the kits or using them for their own R&D purposes, then they're more than happy to help on that. As I mentioned, we've developed a smaller patch which is used for the infants. So this will be this is okay down to the age of 1 month, and we've now tested this in a study. So yeah, really happy to somehow collaborate with anyone who's interested.
Thanks very much. I think that's all the questions in the chat, but if anybody has any questions that they, oh, hang on, there's one more in the Q&A. There's a question from Jeff. The speakers have described how the vaginal microbiome changes with age, particularly through the perimenopause. How does it change with ethnicity, if at all? And is there any intrinsic or extrinsic factor like diet, location, etc.?
Yeah, so that's a question I often wonder the answer to as well, because it is so distinct between the different ethnicities. But I think it must be a combination of factors, like so many things that are complicated in life. I think there could be genetic components, there could be just location, exposure, passed from mother to child, on and on. Diet, of course, could play a role, even in the vaginal microbiome, and just any other number of like locality factors, but that doesn't explain it all, because we see these differences not just based on where you're living. So there have to be other genetic factors as well.
Thanks. And we've maybe got another 10 minutes or so. I'd like to invite the speakers and the panel members to perhaps project yourself forward 10 into the next 5 years. Where do you think we'll be with you know, skin microbiome research, feminine health? In terms of the commercial side, we heard first thing from Rachel what the market is looking like. There are a load of new categories appearing. How can the research community work with the businesses to evolve feminine care? So perhaps Kelly, do you want to give a minute on that? And I'll move around the table, so to speak.
Sure. I think for the consumer, it's going to look like there's new options available to women to have better, I'll just focus on vaginal microbiome, for better care there, for more education of practitioners, doctors on things beyond just what they're taught in med school, how things have changed now. And I hope there'll be an awareness among women that they can get tested, they can find answers to their recurrent problems, and that they're being taken seriously in research and not left out.
Yeah, mute.
Oh, you're muted.
How about for Perfectus Medicus, how do you think that the situation is going to change over the next few years from your business is concerned?
Well, I was going to mention, just related to, first of all, from women's health, all of the advancements around even non-hormonal contraceptives, non-antibiotic alternatives for vaginal health. I mean, those are all being worked on and developed, and are even in some of the further phases of clinical trials. I think that's really exciting, because you know, even taking hormonal contraceptives, how does that affect your vaginal microbiome in the first place? And then I think the combination of the diagnostics for the personalized health is important. For us, it's continuing to combine the research strategies that can predict the clinical efficacy as much as you can. And so understanding the complexity that's happening in the clinical setting through all of the diagnostics that we are, all the data that's coming in, and then being able to come back in and replicate that in the laboratory, such that you're basically going, you know, non-live animal alternatives straight through.
And David, from your point of view, the Skin Trust Club, I have downloaded the app. I'm kind of quite nervous as to what to do next, but do you think that the Skin Trust Club is going to open up this whole area to more consumer related understanding and to give a much bigger data set about,
we hope so.
the human conditions?
We are trying, yeah, what we're trying to is to democratize this ability to look at data, to have more information with your skin. So people spend thousands of pounds per year looking at cosmetic products, cosmetic creams, without really knowing what they're doing to their skin, just subjectively thinking that, oh, this looks at me. And we are very, very surprised. For instance, one of the things we are looking at is how people thinking of their skin is most of the time wrong with the data we are collecting. So when we ask you, what type of skin do you think you have? So 80% of the time people say something which is completely different with the data we are getting. Which is interesting. So, and the microbiome is underused in this sort of thing. So it can be used as a biomarker. It is well known as Oliver was showing this. It's a link between eczema or atopic dermatitis and the microbiome. Even in psoriasis, which is not a clear link, still you can use the microbiome changes as a biomarker that tells you things about the severity of your psoriasis and things like that. So this is going to be used more and more. But also you can add layers of information. We're talking about microbiome. You can analyze the genomics from the person. You can do metabolomics. You can do transcriptomics. And you can add all that layer of information with the ability to look at huge amount of data and AI modeling. And that is an insight that if you do it at the right cost, for the consumers, which is still some time to go to make it at the right cost, it will change our ability to look at our health forever.
Very good. Julie, how about you from research?
Well, I guess I probably shouldn't have been surprised at how little there was on the menopause and the skin microbiome, and I think really we need to drive changes in the curriculum, particularly for medical students, because they have very little knowledge about the impact of the menopause on women, and I think it's kind of almost been a taboo subject for such a long time that's been sort of brushed under the carpet and not talked about. And I think people are talking about it now, and that's a great thing. And I think because women are spending, almost half their life in a state of estrogen deficiency, because many women now are living well into their 80s to 90s, so they've had sort of half their life without estrogen, and I mean certainly in older ladies the incidence of UTIs is very high, and because the vaginal epithelium, like the skin epithelium, is so thin, and the tissue, I mean when we look at the histology of the skin, we can just see the huge change in the architecture of the skin with aging. So, and I think again having an understanding of estrogen biology is really important, because there was such a lot of scaremongering with the HRT that it was going to cause breast cancer, and of course that's not, but people are still very reluctant, and GPs are still very reluctant, physicians are still very reluctant to have anything to do with any kind of estrogen therapy. So I think we need a lot more education in that.
And our three other panelist members, Ryan, from your perspective, how do you think the research is going to develop over the next few years?
Well, so when I first started looking at vaginal infections, it was looking at recurrent vulvovaginal candidiasis, and we went back and we did a retrospective study of some clinical isolates which were in a hospital, and I was really surprised to see that many of these were just called yeast. These weren't classified any further than the genus level in many times, so they're just Candida. And what you have is a significant portion, in terms of yeast anyway, you've got a significant portion of these will be non-albicans yeast, which are intrinsically resistant to the frontline antifungals. And these are, again, another problem in terms of recurrent vulvovaginal candidiasis is that these are readily available over the counter. So actual antifungals these antifungals don't need a prescription, which has the obviously the things like promoting antifungal resistance. All these things are some problems with that. So the kind of recurrence of the infections is something which I think is very interesting, trying to get some sort of treatment or therapy which will be able to stop. So not just a stopgap for a few months and then you get a recurrent episode. So being able to fully resolve the course of the infection is something which I think is an area of interest.
Right. Oliver, collecting samples is obviously creating large amounts of data. How do you think, how do you think you can utilize the data you're collecting usefully for, the wider popular use in the future?
Yeah, I think to everyone's point, what everyone is hinting towards is this personalized medicine approach and how diagnostics is advancing. I mean, that's interesting, Ryan, you're saying that they would only put down the genus level. And I see in the future, it will be down to the strain and substrain, and it will be so personalized and targeted, both in, if we were talking vaginal microbiome, skin microbiome. I think an ease in which the consumer or patient can take these tests at home is going to be really important. And so what we're trying to do is make that process as easy as possible, in using a patch, trying not to be too clinical, but collecting information that's really important. We've collected thousands of samples now, both from Asia, from Singapore, and from the US. And so we're seeing some interesting things. Also a similar app approach where they can have a look at their report through an app and see their skin health. And so the more data we collect and understand these correlations between their current health, their diet, their environment, is going to be really important to making these links between what are these biomarkers and how can we kind of interfere with them. But yeah, I think it's a really exciting future for the space that we're all in here.
Excellent. And Chiara, last word to you, as a very young entrepreneur in this area. You've talked about some of the challenges that you faced starting a business, but how do you think the future looks for products that are based on interacting with the human microbiome?
Well, I think it's an exciting time, I think, for the microbiome. I think more and more investors are now interested in the space, and I think also female health, I think now more people are seeing could be also a great, potential business business case for a lot of investors, because it's obviously like a huge market, which I think has been quite overlooked. And so I think now new ideas are coming to the market. So I think there are a lot of exciting opportunities. And I think also adding a bit to what everyone said, I think really also destigmatizing the conversation around it and making solutions that are really accessible as well, what Oliver just said with the patch, and really listening to consumers. And I think kind of offering solutions that are easily accessible for women, I think is also massively helpful in this space, and kind of trying to make women aware of making this part of the routine and how you can make it part of your routine and making it normal. I think it's a really important aspect of the educational bit, and also then looking at this, why we're here today, and I think working together and working on cross-promotional activities as well, like how can we work together in order to address this, because it's so connected. I think it's a very important aspect.
So thank you very much, everybody. Katerina, have you got anything that you want to add at the end?
Yeah, I guess I have some farewell and thank yous Thank you. Some last words. So I would like to thank the speakers for truly interesting talks. And really what's important for me is high quality of translational science. Also very much thank you to the NBIC and CCUK team, which are behind us to make this happen. I think this is an exciting kind of space. There is exciting future in terms of the science and the translation and the consumer attitudes. As you said, these are changing. This webinar will be followed up with an event to aid BIT funding development. We would like to create a community, both of the academics and the SMEs and the larger companies, David. So to kind of help, perhaps the academics call it the sandpit, but to look at where funding would come from, where is the investment coming, you know, what the investors are saying, to create a community. And again, that will be led by CCUK and Bradford University. So I hope you enjoyed yourself today, and thank you all for coming to the table, be that short and sweet for you, Chiara, Oliver, and Ryan, and we hope to work with you again. Thanks, everybody.
Yep.
Thank
you.
for organizing.
Thank
you all.
Thank you.
Bye.
Bye.